Understanding pests and diseases. The first step, how to identify the common kharif pest means pests nothing but insects, mites, nematodes, rodents, bugs that cause damage to crops. In the pest, there are several pests like sucking pests, chewing pests, and other pests. A sucking pest nothing but aphids, jassids, whiteflies, plant hoppers, thrips. How to identify the sucking pest? Looking for yellowing, curling, or distortion of leaves, stunted growth, presence of sticky honeydew, leading to sooty mold, and direct observation of small insects on the underside of the leaves or tender shoots. Damage, they suck sap from plants, weakening them, and can transmit viruses. Example, aphids on leaves can absorb the yellowing, curling, as well as digestion of leaves, as well as stunted growth and presence of sticky honeydew, leading to smoothie mold. Next, chewing pest. Chewing pest nothing but stem borer, leaf folder, army worms, bollworms, grasshoppers are the chewing pest. How to identify is looks for bore holes. We need to check the holes in leaves, cut stems. First, the pest make a hole in leaves or cut stem. Skeletonization of leaves will be takes place while chewing pest need to be identified. Then only chewing pest we can be observed. ఫస్ట్ హోల్ చేస్తుంది ఆకులల్లో పత్రాలకు కాడల్ని కాండంని కట్ చేస్తుంది జల్లెడ పడుతుంది లీవ్స్ని స్కెల్టైజేషన్ మీన్స్ బోర్డ్ ఫ్రూట్స్ స్టెమ్స్ డిఫోలియేషన్ లీవ్స్ రాలిపోతాయి presence of caterpillar and larvae majorly chewing pest in the presence of caterpillar and larvae can make huge damage is direct feeding on plant parts leading to yield loss or plant death. Example of chewing pest is stem borer majorly. Other pests, termites, root grubs, rodents. How to identify wilting of plants despite of adequate moisture, damaged roots, nothing but digging up a plant, gnawed stems, and burrows in the field. Wilting of plants despite of adequate moisture. తేమ శాతం ఎంత ఉన్నా కూడా ఆటోమేటిక్గా చెట్టు ఎండిపోతుంది దాన్ని విల్టింగ్ సిమ్టమ్స్ అంటారు అది పెస్ట్ ద్వారా రూట్ గ్రబ్ ద్వారా వచ్చే అవకాశం ఉంటుంది టర్మైట్స్ రూట్స్ termite root grubs rodents also causes gnawed stems and burrows in the field rodents. Next, identification of common kharif diseases. Here after the pest we need to identify another step in the management is main disease caused by pathogens like fungi, bacteria, virus, and nematodes. Fungal diseases like blight, rust, downy mildew, powdery mildew, leaf spots, mudds, and damping off. Here, how to identify these diseases? Blight. When the blight appears on the leaves, rapid browning and drying of leaves will be symptom, and the stems and flowers also. Sometimes blight will be occur in severe stages. Rust. How to identify the rust is nothing but powdery rust colored putule, nothing but spots, rust colored pustules on leaves and stems. Here another diseases is downy mildew and powdery mildew. Here mildews nothing but white, powdery mildew is nothing but white colored, nothing but powdery mildew. Downy or downy mildew on growth on leaves. Downside of the growth of the leaves with the powdery mass is called downy mildew. Leaf spots, circular or irregular spots on leaves is nothing but leaf spots, often with distinct margin or halos. Smuts, nothing but black smoothie mass replacing grains or plant pots. Damping of nothing but Diseased seedlings collapsing at the soil line, common in nurseries. Damping of is common in nurseries. Here, rice blast disease you can observe, and downy middle on maize you can observe. Bacterial disease, next bacterial blight and bacterial wilt. How to identify this? Water soaked lesions, wilting of leaves despite acquired adequate water, streaks on stems, and foul smell is nothing but bacterial disease. Viral disease here, mosaic leaf curl, yellow bean mosaic is the famous viral diseases. How to identify is molting, yellowing, curling, distortion of leaves, stunted growth, vein clearing, and banding. Often spread by sucking pests. Sometimes sucking pests also spread viral diseases. Nematode diseases, root knot nematodes. How to identify root knot nematodes? Galls, formation or swelling on roots, stunted and yellowed plants, reduce yield. Practical tip for identification. When we are going for the field regularly, we need to walk through the field, observe plants closely. Hand lens. Use a hand lens to see small insects or disease structures. Reference materials. Keep pictures or samples of common pests and diseases for comparison. Expert constellation. Expert consultation is important. If unsure, take samples to an agriculture expert or university lab. Another thing is integrated pest and disease management. IPM or IDM, the holistic approach we need to take up. IPM is nothing but integrated pest management is a sustainable approach that combines various methods to keep pest and disease population below economic injury level while minimizing the harm to the environment and human health. Practical IPM strategies for kharif crops are cultural practices, mechanical or physical practices, or biological practices is been going on. Cultural practices, nothing but prevention is better than cure. Field sanitation demonstration show how to remove and destroy crop residues after harvest. We need to go into the field and walk through, and we need to remove and destroy the crop residues after harvest. Explain that this is eliminates breeding grounds and overwintering sites for pests and disease spores. Concept removing infected plants or plant parts from the field. Crop rotation. Discuss and illustrate a simple crop rotation plan. Planting a non-host crop after a host crop. Explain how it breaks the life cycle of soil-borne pests and diseases. Resistant varieties. Demonstration. If possible, show different varieties of a kharif crop, highlighting which one are known to be resistant to certain common pests and diseases. Explain how using resistant varieties is a primary and cost-effective defense. Proper planting and timing and spacing is another cultural practices. Here we need to demonstrate to correct spacing between plants and rows. Explain the optimal spacing improves air circulation, reducing fungal diseases, allows for better light penetration, improves the importance of timely sowing and planting to avoid peak and pest disease pressure. Balanced fertilization and irrigation. Demonstration. Explain the concept of giving the right amount of nutrients and water. Too much nitrogen make plants more susceptible to sucking pests and some diseases. Over irrigation can create humid conditions favorable for fungal diseases. Weed management demonstration. Show some kharif weeds and explain how they can act as an alternate host for pests and diseases. Demonstrate manual weeding or discuss herbicide application. Mechanical or physical methods. Direct removal or barrier creation. Hand picking. Here demonstrate hand picking. How can we demonstrate means in small plots demonstrate picking large caterpillars or diseased leaves or fruits by hand and destroying them. Pheromone traps. Demonstrate set up pheromone traps lure specific male insects in the field. Explain how they help monitor pest populations and can reduce mating. Monitor pest population can reduce mating. Sticky traps, yellow or blue. Place yellow or blue sticky traps in the field. Explain how they attract the trap small flying insects like whiteflies, aphids, and thrips. Help in monitoring and mass trapping. Light traps demonstrating, if available, show a light trap attracts nocturnal insects. Explain its use for watering and sometimes reducing the pest population. Bud patches. Demonstration install bud patches in the field. Explain that buds are natural predators of small insects, pests. Biological control using natural allies, predators, and parasites. Demonstrate, if possible, show pictures or even specimens in controlled environment of common natural enemies. Ladybug, bugs feeding on aphids, lacewing larvae. Explain the role in controlling pests. Concept releasing or conserving beneficial insects that prey on parasitizing pests. Biopesticides are bioagents. Demonstrate, show examples of commercially available biopesticides like trichoderma for soil-borne diseases, Bavaria basana for insect pests, Bacillus thuringiensis for caterpillars. Explain how they are living organisms or the products. Control pests and diseases. Application. Demonstrate safe mixing and spraying of biopesticides. Example, farmers spraying biopesticides. Chemical control. Last resort. Use it judiciously. Threshold levels concept. Explain the threshold, economic threshold level, ETL. The pest and disease population density at which control measures should be initiated to prevent economic damage. Emphasis that chemicals are used only when other methods are insufficient and pest and disease population reaches economic threshold level. Safe handling and application. Crucial, demonstrate the proper way to handle and mix apply pesticides wearing PPE, gloves, mask, goggles, protective clothing, explain dosage, timing, and spraying techniques, covering the underside of the leaves for sucking pests. Briefly explain different types of insecticide, fungicide, herbicide, and their mode of action. Targeted application concept. Emphasize applying pesticides only to be affected area or plant parts. Herbicides and their modes of action. Minimize, and the herbicide concept should be minimize the environmental impact. Rotation of chemicals. Importance. Example, explain the importance of rotating different chemical groups to prevent pesticide resistance in agriculture. Practical class activities demonstration. First should be field walk and observation. Take students who crop field them to identify different plant parts, root, stem, leaf, flower, fruit. Activity, look for symptoms of pest damage, holes, yellowing, webbing, presence of insects. Activity, looking for symptoms of diseases, spots, wilting, powdery growth, discoloration. Encourage them to collect samples of damaged plants or pests under supervision for closer examination in the lab. Microscope or a hand lens examination in the lab. Examine collected samples under microscope or with a hand lens to see details of insects, fungal spores, and bacterial use. Preparing bioagents, showing how to prepare trichoderma every enriched farmyard manure, how to dilute a liquid biopesticides for preparing a bioagent. Sprayer calibration, if time permits, demonstrate how to Calibrate a sprayer to ensure correct dosage application. PPE demonstration. Have all necessary personal protective equipment on display and demonstration how to wear and use it correctly. Case studies on discussion activity. Present scenarios of common kharif crop pests or diseases problems. Ask students to propose IPM strategies. Seed treatment. Demonstrate how to treat the seeds with fungicides or bioagents before sowing to protect against soil-borne diseases. By combining all theoretical knowledge with practical demonstrations and activities, students will gain a solid understanding of pests and disease management in kharif crop production. The key is to emphasize observation, identification, integrate natural pest and disease control, moving away from soil reliance on chemical solutions. Please like and share and subscribe. Jai Hind.